A very warm welcome to all of you guys. So welcome back in another video tutorial from microbiology with Shagni. So in this video tutorial, we will going to extend our discussion of Kelvin cycle with the help of another important topic, that is photorespiration. So here we will going to learn that not only CO2 but oxygen also can bind with ribulose bisphosphate carboxylase. But in normal condition, it cannot compete with CO2. Okay, but when the environment become hot, dry, and less moist is present in the environment, the concentration of the CO2 starts to drop, and as a result, O2 can get the opportunity with to bind with RUBP carboxylase. Okay, so what's the idea? The idea is during hot and dry condition, the pores of the leaves, because remember in the leaf of the plant there are various type of pores present which we are calling as stomata. This stomata will going to close. Okay. Now the question is why they are closing the pores of the leaves. The actual idea is the plants, when there is a very short storage of water, especially in the tropical arid areas or semi arid areas, during that time the plant try to preserve water inside it, and that is why they are closing the pores of the leaves. Okay, and as a result the CO2 concentration starts dropping. Okay, and when the CO2 level drops below 50 parts per million, the ribulose bisphosphate carboxylase starts fixing O2 instead of CO2, and this is the photorespiration. Okay, and as a result, what will happen? Obviously, RUBP will going to react with oxygen instead of CO2 with the help of the enzyme, and as a result, two things will be generated. One is 3 carbon containing 3 phosphoglycerate, which was also the compound for our CO2 fixation, as well as 2 carbon containing 2 phosphoglycolate. Okay, so that's the idea. That is what we call photorespiration, and that is why we are giving RUBP carboxylase the name Rubisco, that is RUBP carboxylase slash oxygenase. This is the oxygenase reaction that we are talking about in our previous video, right? So why we are talking about the photorespiration? Is this very essential and good for plant? So the answer is no, because it reduces the efficiency of CO2 fixation by as much as 50%, and as a result, obviously the carbohydrate production will also be dropped, right? And this is not good because carbohydrate is the nutrition that plant requires, right? But plant also have to preserve water for its own survival, and that's why they have to drop the CO2 level, right? So that is the thing. So there are some plant that is known as C3 plant that especially rely on the RUBP carboxylase for the fixation of carbon dioxide. For them the photorespiration is especially very bad, but there are some plant designated as C4 who have the extra ability to fix carbohydrate with the help of an alternative pathway, and obviously with the help of an alternative enzyme known as PEP carboxylase, phosphoenol pyruvate carboxylase, that will going to fix carbon dioxide with PEP, producing 4 carbon containing oxaloacetate. Okay, and in that type of reaction this oxaloacetate will be converted into other 4 carbon containing intermediate, and that's how they can transport CO2 into the photosynthetic cell. Okay, and minimizing photorespiration. Okay, so this type of pathway is known as Hertzsprung pathway, and the plants that perform this type of pathway are known as C4 plants. So before that, this is the reaction. Phosphoenol pyruvate will going to react with HCO3 minus, giving us oxaloacetate. Okay, so Hertzsprung pathway is usually occurring in case of plant who have the ability to use PEP carboxylase for carbohydrate fixation, especially in tropical arid areas and semi-arid places. Concept is the same, to prevent the loss of water, stomata is closed, and that is why carbon dioxide level starts dropping, and as a result RUBP carboxylate starts fixation of O2, that is the photorespiration, but it reduces the efficiency of carbon dioxide fixation, and that is why the plant tries to minimize the photorespiration, right, with the help of another pathway, right, that is the alternative reaction to trap CO2. Although it is not a CO2 fixation, and although it is not an alternative of Kelvin cycle, but it is a CO2 delivery system to the interior portion of the plant, okay, where it cannot, or where O2 cannot compete with CO2, okay, and as a result CO2 can easily bind with RUBP carboxylase for the carbohydrate production. Okay, that is the main goal of Hertzsprung pathway. Again I am telling you, it is not a CO2 fixation or alternative of Kelvin cycle, it is just a CO2 delivery system to directly transfer CO2 into the inner part of the plant where CO2 cannot fight with it. Okay, sorry, not CO2, where O2 cannot fight with it. Okay, so that RUBP carboxylase can easily use it, right, for the carbohydrate production, and that's how they try to minimize the photorespiration. Okay, so oxaloacetate and malate are the CO2 transporter that mainly involved in this CO2 transportation. Okay, so that's how C4 plant try to maintain a high CO2 concentration over O2 concentration, and that's how they minimize the photorespiration. Okay, so although C4 plant is very beneficial, especially in dry condition, heat condition, right, but when there is a moisty condition or less sunny environment, C4 plant simply cannot fight with C3 because C3 have a active Kelvin cycle. Okay, that's why C3 will going to fix more amount of CO2 as compared to C4 when there is a moisty environment, right? But opposite will be when there is a hot or dry environment. Okay, so that's the idea of our C4 pathway or Hertzsprung pathway. That's how a additional CO2 delivery system helps plant to minimize photorespiration. Okay, so that their carbohydrate production will not be reduced drastically. Okay, so that is the idea of our C4 pathway or Hertzsprung pathway. So that was our second video of Kelvin cycle. So I hope you have learned it very well. So thank you for listening to this class.